Good morning, everybody. If you're wondering why there's so much recording equipment, it's not that my lectures are so good that they're being transmitted live to CNN or something like that. But well, you might have noticed that there's a camera installed in here and next door. So I'm just testing whether we actually get a good recording of this lecture with this built-in camera, so we can start using that instead of the ones that we carry around. So that's the only reason. In today's lecture, we should be talking about the synthesis and degradation of neurotransmitters. So it's going to be, once again, after a little break, a lecture focused on metabolic pathways. But obviously, there's going to be a lot of other things. It's not just going to be metabolism, but it's going to be other things that I'm going to be talking about. In the previous lecture on synaptic transmission of information in the nervous system, I mentioned most of the things that I will now just recap briefly. Most neurotransmitters, almost all neurotransmitters, are synthesized in the synaptic terminal, in the presynaptic neuron. What is the only exception to that? Which neurotransmitters are not synthesized in the synaptic terminal? Which chemical neurotransmitters are not synthesized in the synapse itself? Huh? I think we mentioned last time.
Proteins and peptides.
Excellent. Okay, proteins, peptides, because they need to be synthesized in ribosomes, so they're synthesized in the nerve cell body. Everything else is synthesized in the synaptic terminal. So neurotransmitter is synthesized. It's packaged into secretory vesicles. These are released by the mechanism that we talked about. They get into the synaptic cleft, where they activate postsynaptic receptors. And in order to terminate the signal in the synapse, we need to get rid of the neurotransmitter that's been released. And there are two ways of doing that. One is degrading the neurotransmitter itself, which is the case for acetylcholine, okay, typically. Some other ones as well, which I'll mention. And the other way to do it is to pump the neurotransmitter away from the synapse. So either pump it back into the presynaptic neuron, which is called reuptake, or it can be taken up by some cells sitting around the synapse, which are usually astrocytes. Okay? So some neurotransmitters are pumped into astrocytes, and then something happens to them. Okay? So that's the general overview, and we're going to be talking in more detail about the individual steps in this general picture. One more thing to keep in mind when we're talking about neurotransmitters, the blood-brain barrier, which you've heard about the structure, is impermeable to practically all fully formed neurotransmitters. So neurotransmitters that may appear in the blood, for example, as hormones or something else, they will not get into the brain. The brain cannot take them up or anything like that. That obviously makes sense because we don't want to be mixing up signals that are going on in the brain and that are going on elsewhere in the body. So the blood-brain barrier actually is impermeable to fully formed neurotransmitters. So they need to be formed in the nervous tissue itself. And again, we're going to be looking in detail at the individual steps in these pathways and how it's done. Do you have any questions about the general picture? Yeah, that was mostly just a recap from previous lectures. So let us first talk about the two most important, the most abundant neurotransmitters in the central nervous system, which are serotonin, dopamine. GABA is one of them. Not acetylcholine. Not dopamine. Glutamate. Glutamate and GABA account for more than 90% of all synapses in the central nervous system, probably substantially more than 90. Okay? So almost all synapses in the central nervous system, especially in the brain, have either glutamate as a neurotransmitter or GABA as a neurotransmitter. Okay? So all the other ones that you mentioned and all the other ones that we're going to spend substantial amount of time talking about their metabolism, they're actually minority neurotransmitters. They're very important for the proper function of the brain, but as far as abundance goes, they're just tiny, tiny minority. Okay? So the most important ones are glutamate and GABA. Right. Let's start with glutamate. Okay, this is glutamate. How do we make glutamate? By transamination of what? Excellent. We get it by transamination of alpha-ketoglutarate. And where do we get alpha-ketoglutarate?
Krebs cycle, for example.
Okay, it's an intermediate in the Krebs cycle. Okay, a very simple synthesis in one step by transamination from alpha-ketoglutarate that we get from the Krebs cycle. Okay, simple synthesis, but we'll see in a minute that neurons actually do it in a slightly different way. But before we talk about that, let us make GABA. Does anyone know how GABA is synthesized? Or what is the, from butyric acid. Okay. Well, this is GABA. So what do you think? How do we synthesize GABA?
Decarboxylation of glutamate.
Excellent. That's exactly how it works. Okay, we decarboxylate glutamate and we get GABA. What do you think the enzyme is going to be called? What is going to be the name of the enzyme? GABA carboxylase. Okay, so going in the opposite direction. Glutamate decarboxylase. Okay, glutamate decarboxylase. A very important enzyme. You should remember it. Okay, glutamate decarboxylase makes GABA. So that's the chemistry, and there is relatively little that's interesting about that. It's going to be a little bit more, there are going to be some more interesting things about degradation. But let's have a look how it actually works in the central nervous system, in the neurons and other cells. Okay, I'm gonna, yeah. So in order to see the complex metabolism of GABA and glutamate and how it actually works in the nervous system, I'm going to draw a schematic that's going to have two synapses. One of them is going to be a glutamatergic synapse. So it's a synapse that uses glutamate as a neurotransmitter. And the other one is going to be a GABAergic synapse. And what we're going to have in the middle is an astrocyte. Now, this is just a scheme so that I can put everything in one picture. I'm not saying that each two synapses, always a glutamatergic and a GABAergic, always have one astrocyte connecting them, okay? It's just a scheme, okay? It's just a picture to explain things, okay? Good. So let's start with the glutamatergic neuron, or the synapse. Now, we have glutamate in the secretory vesicles. Okay, let's now forget how it's made, because we're going to see it in due course. Once the synapse is activated, once the presynaptic neuron is activated, it releases glutamate into the synaptic cleft. And glutamate, after it's there, it activates some receptors here, as usual. Okay, but we need to get rid of glutamate from the synaptic cleft. And you already know how we do that.
Decarboxylation.
Yeah. Most of it, no decarboxylation. Most of it is taken up by the astrocyte. There's a specific transporter there that pumps glutamate away from the synapse into the astrocyte. There's a little bit of glutamate that goes back into the presynaptic neuron by reuptake, but the majority pathway of getting rid of glutamate is through a specific transporter into the astrocyte. Okay? Now what happens with glutamate in the astrocyte? There, it's turned into another amino acid, which is glutamine. Okay? Glutamate into glutamine. How do we do that? How can we make glutamine from glutamate? Transamination. We can't really use transamination because glutamine is not an amine, it's an amide. It's also an amine, but...
I remember we learned it in metabolism because it was used to transport nitrogen
Yes.
through the blood.
Very good, yes.
I can't remember the name of the enzyme though.
Okay, that's fine, but what do we need for that? So you said that glutamine is used to transport nitrogen in the blood. That's correct. So what kind of... So we need some... Probably need some nitrogen to add to glutamate to make glutamine, yeah?
Is it amine group?
It's...
You attach an amine?
That's true. Glutamine is an amide. Yeah, it has an amide group. But how do we make it?
Nitric oxide?
Nitric oxide?
Yeah,
No. We actually use ammonia. Okay? We use ammonia to make glutamine from glutamate. But we need one more thing for that. No? Okay, here you can see that it's actually very useful to know the structures of amino acids. Okay? Because the difference between glutamate and glutamine, so this is glutamate, and glutamine is this. So we definitely don't need any bicarbonate for that. We only need ammonia. But the other thing we need is energy. We need ATP. The enzyme is called glutamine synthetase, a fairly important enzyme in metabolism and in the brain as well. Okay, so from glutamate that was taken up from the synapse, we made glutamine using ammonia and ATP. Now, this glutamine is then exported into the neuron and then recycled to glutamate by an enzyme called glutaminase. Okay, so now we have a nice cycle starting with glutamine, changed into glutamate, released into the synapse, taken up by the astrocyte, turned into glutamine again, re-exported into the neuron.
Sorry, can you
Yep.
repeat the enzyme that does that?
This is glutaminase, so an enzyme decomposing glutamine, glutaminase. And this is glutamine synthetase. Okay? So that's one half of the system. Let's now have a look at the GABAergic synapse. So we start with GABA. We release it into the synapse. And what do you think happens to GABA released in the synapse? Yes. It's taken up into the astrocyte. Again, there are transporters for that. So now we have GABA in the astrocyte. Now the astrocyte needs to do something with this GABA. It can't just recycle it, because even here the recycling is not done with glutamate, is it? Because the astrocyte can't just pump out glutamate, because that would screw up the signaling in the brain. Do you see that reason? Yeah? It needs to turn glutamate, which is a neurotransmitter, into something that's not a neurotransmitter, but can be turned into a neurotransmitter. Okay? So we turn it into glutamine, pump it back. Here, GABA needs to be turned into something else that's not a neurotransmitter and can be recycled into GABA. Guess what it could be? It can't be glutamate, because glutamate is a neurotransmitter. But you're very, very, very close. To glutamine. Okay? So in the end, we turn GABA into glutamine, and we pump it back into the presynaptic neuron. Here, it's turned, glutamine is turned into glutamate, and glutamate is turned into GABA. Okay? Do you understand this bit?
Sorry, is it glutamate synthesized out of the astrocyte or?
So GABA takes up, the astrocyte takes up GABA. By some steps that I'm going to describe in just a second, it's turned into glutamine. Glutamine is exported back into the presynaptic neuron, and then it's via glutamate, it's turned into GABA again. Now, let's talk about these missing steps here. Okay, this is GABA. What can we do with it? We're going to need several steps in order to turn it back into glutamine. Any suggestions what we could do with this molecule? Carboxylation. Okay, that's a very good idea. Basically putting back the carboxyl group and making glutamate. Very good idea. Unfortunately, that would be too easy. So that's not what we do. Okay, first of all, we transaminate this amine. There's transamination going on, okay? The enzyme is called GABA transaminase. And what do you think is going to be the product of this transamination? What's going to happen to this amino group? That's all I'm asking for. Huh? It's going to be removed, and what's going to be there instead of what group is going to be left behind that reaction? Not a carboxyl group. An oxo group. So in this case, it's going to be an aldehyde. Okay? Do you see that? In normal transamination, because it's a secondary carbon, what we get is an oxo group, and in this case, it's a keto group, usually. But here, because it's a primary oxo group, it's an aldehyde. Like that. Yeah. Okay? We get an aldehyde group. What can we do with this aldehyde? Does it look like something? Does it resemble some other chemical that might be an intermediate in some very important metabolic pathway? Huh? Alpha-ketoglutarate, not really. Oxaloacetate, there's even something that resembles even more closely. Malate, not really. I think I've heard it. Succinate. Okay? In one simple step, we can oxidize this aldehyde into carboxyl, and we get succinate. Okay? This compound is called succinate semialdehyde. So it's an aldehyde, only we take succinate and we turn one of the carboxyls into aldehyde, so it's a semi-aldehyde, just half aldehyde. Okay, succinate semialdehyde. It's not that important to remember that. But in one step, we oxidize the aldehyde, we turn it into succinate, and once we have succinate, we're fine, because we can easily make glutamine. How do we make glutamine from succinate? Okay, which pathway is succinate part of? Krebs cycle. Okay. Can we use Krebs cycle maybe to make glutamine in the end? Okay, how? Any from oxaloacetate? Okay, so yep, you're right, there is oxaloacetate in the Krebs cycle. So what we do, we take succinate, we turn Krebs cycle by about 1/2 turn, so we add acetyl-CoA, we make oxoglutarate, we transaminate oxoglutarate to glutamate, we add another amino group, and we have glutamine. Okay? So what we do here is we turn it to succinate semialdehyde that goes into mitochondria, is turned into succinate. There is the Krebs cycle, so it spits out 2-oxoglutarate, which we then transaminate into glutamate, and we aminate it again to make glutamine. Okay? GABA transaminated into succinate semialdehyde. That's turned into succinate. By 1/2 turn of Krebs cycle, we get 2-oxoglutarate. From 2-oxoglutarate by transamination, we make glutamate. And then by amidation, which we've seen already here, we make glutamine and we pump it back into the neuron. Okay? So that is the metabolism of glutamate and GABA. Now, a couple more remarks about this whole glutamate-GABA system. One important thing to realize again is, as I said, no fully formed, practically no fully formed neurotransmitters are capable to move through the blood-brain barrier. Glutamate is one of them. Glutamate does not cross the blood-brain barrier. And there are several reasons for that. One is that under normal conditions, the concentration of glutamate in the brain is much higher than the concentration of glutamate in the blood. So there is no reason for glutamate to get from the blood into the brain. Okay? Yeah, it's more glutamate in the brain, so it won't flow from the blood. Also, the transporters in the blood-brain barrier are set up in such a way that they basically suck glutamate from the brain into the blood and not the other way around. Okay? I'm not going to go into details, but basically glutamate under normal conditions does not get from the blood into the brain. It needs to be made metabolically inside the brain. Okay? So you may have heard about the problems of some people that eat glutamate in Chinese food. It's called Chinese restaurant syndrome. Not sure if you've heard about it. The idea used to be that it's this flood of glutamate that gets into the brain and obviously disrupts all these regulatory networks. That doesn't seem to be likely, because the amount of glutamate that a person would have to ingest would be just insane in order to get the blood concentration of glutamate so high that it would actually turn around the transport. So there are probably other reasons for what is called a Chinese restaurant syndrome. The other thing that I want to mention, which is a very important clinical correlation of this whole system, there are conditions where the level of ammonia in the blood rises above normal values. It's called hyperammonemia. It's just a descriptive term. Hyperammonemia. Would anyone try to venture a guess which conditions could cause hyperammonemia? The primary organ, yeah?
Liver failure?
Very good. Do you want to explain why liver failure would?
The urea cycle takes place in the liver, so if you stop that, then you get more ammonia.
Excellent. Okay. The liver is the primary organ responsible for removing all sorts of nitrogen, including ammonia. Also, a lot of ammonia comes from the gut, from the intestines, because the bacteria that live there produce a lot of ammonia. So the liver is the first filter through which the blood from the gut goes. So it's responsible for removing most of the ammonia. Now, if there are liver problems like cirrhosis or acute hepatitis or something like that, the liver may not be able to do that, and the level of ammonia starts rising. Now, the first organ that starts suffering is the brain. Can anybody try to explain why the brain would suffer when there's too much ammonia in the blood? Yeah?
It would push the equilibrium towards glutamine, so you can't create GABA in that presynaptic neuron to the right there.
Oh, here. Here, that basic...
No, yeah, I was thinking about that one.
Okay.
Yeah.
Isn't it the same thing? But okay, fair enough. So instead of making glutamate from glutamine, it would be making the opposite, or... Okay?
But, I mean, you said it was a neurotransmitter, so if you've got too much glutamate, then you've just got signals happening all the time. So
Okay.
Isn't that the reason? Or?
But why do we have too much glutamate?
Because we can't get rid of it.
Okay, yeah? Sorry, who was, yeah?
Ammonia is capable of crossing the blood-brain.
Yeah, definitely. Okay, so all the things that you said are true. The reason why hyperammonemia causes brain problems, it's called encephalopathy, usually liver encephalopathy, is all these reasons. So ammonia, being a small molecule, uncharged molecule, gets very easily through the blood-brain barrier, so it doesn't need any transporters or anything like that. So if there's high level of ammonia in the blood, there's going to be high level of ammonia in the brain. Now, as you correctly said, a lot of these reactions, which are crucial for proper functioning of the brain, require some regulation and are affected by the amount of ammonia. As you correctly said, the synthesis of various and degradation of various neurotransmitters is dependent on the level of ammonia. And the other reason is that the synthesis of glutamine, which also depends on ammonia and is regulated by ammonia levels, also needs ATP. So there's a lot of ammonia. The astrocytes will start producing as much glutamine as they can, and their ATP becomes depleted. And then they suffer because they don't have enough energy for other things. Okay? So liver encephalopathy is caused by a complex dysregulation of this system by ammonia. Okay? So that's a clinical correlation. It's a very, very important condition. Later, you know, in your 4th or 5th year, you can hear about it because it's a very dangerous problem, a very severe one. Okay, any questions about glutamate, GABA, glutamine in the brain? Yep.
Can you just quickly go through again how you get from GABA to GABA to
Okay. Yep, sure. So we start with GABA. We transaminate it into succinate semialdehyde. Yep. We oxidize it into succinate. Yep. Succinate goes into the Krebs cycle as it is an intermediate. And from the Krebs cycle, instead, we get 2-ketoglutarate, 2-oxoglutarate, whatever you want to call it. What are you used to calling it? Oxoglutarate or ketoglutarate? Alpha-ketoglutarate, okay. Alpha-ketoglutarate, okay? So that's what we get from the Krebs cycle. Yeah? Once we have alpha-ketoglutarate, through transamination, we can turn it into glutamate. Well, and you know how to make glutamine from glutamate already. Yep. All right. Any other question?
Isn't there a negative feedback system for the astrocytes? If they, I don't know, what sort of the ratio ATP that makes it stop producing, using too much ammonia?
There are probably many regulations. So, you know, the astrocyte will know that something is wrong. It will definitely detect low ATP-ADP ratio. It will, you know, detect high AMP, etc. So the astrocyte will know, you know, things are going badly. But the way these metabolic pathways are set up, they're set up in such a way that they seem to be the priority.
Because it's toxic.
What is toxic?
Ammonia.
Well, interestingly, the reason why ammonia is toxic is mostly because it does this. But it probably is toxic on its own, but this is the main effect that you see clinically of toxicity. So it's tricky when you think about why is it set up in such a way that, in liver failure, these things happen. To be honest, I shouldn't be spending too much time talking about this. From an evolutionary point of view, if you had liver failure, you died. So there was no point for the body to be prepared for what to do when there's liver failure. Do you know what I mean? So there are no evolutionary mechanisms how to cope with elevated ammonia levels because you would just die. So, okay? So it's set up in such a way that the brain just can't cope with it. Okay. Right. There are no more questions. Let's move on to the next group of neurotransmitters, and those are catecholamines. Oops. Okay. Now, you've already heard about catecholamine synthesis before, so I'm only going to run through it so that I can make sure that you know what's going on, that you know all the names of the enzymes, etc. Right. What is the raw material? What is the precursor of catecholamines? Tyrosine, or we can actually go one more step, phenylalanine. Phenylalanine, which is turned into tyrosine by an enzyme called, What's the enzyme called? Well, what's the difference between the two molecules? So what's the difference between the two molecules? A hydroxyl group. So the enzyme is going to be called, it's going to be hydroxylase, and specifically phenylalanine hydroxylase. Okay, good. We have tyrosine. What do we do with tyrosine? There's another hydroxylase which is called tyrosine hydroxylase. Very good. And we make what? Correct. We make L-DOPA. What does L-DOPA stand for? Very good. Dihydroxyphenylalanine. Okay, L-DOPA. Okay, what's next? Very good. So we remove the carboxyl group. What's the enzyme going to be called? The enzyme? Dopa decarboxylase. Very good. And we get dopamine. Dopamine. There's another hydroxylation. Okay, good. So there's a beta hydroxylation. Where does it happen? On the beta carbon. Okay, which one is the beta carbon? Huh? The third carbon? 1, 2, 3? Okay, this is the beta carbon. Okay. Very good. So we hydroxylated. The enzyme is called?
Dopamine beta hydroxylase.
Very good. Well read. Yes. Dopamine beta hydroxylase. what do we get? Norepinephrine. It's preferable now, generally, to call it noradrenaline. Okay? So norepinephrine used to be the American way of saying it. No, but nowadays the standard bodies say that noradrenaline is the preferred name. Okay? I don't care if you call it either, but it's better to call it noradrenaline. Okay? And the final step. Methylation. Okay, whereabouts? N-methylation. So where is it going to happen? Okay, on the nitrogen. Very good. What is the name of the enzyme? Okay, it's an N-methyltransferase, but there is another bit of the name there. Huh? The full name is phenylethynylamine and methyltransferase. Okay, that's a difficult one, so if I were you, I'd look at it. Okay?
Could you repeat that, please?
Okay? Phenylethynylamine, and methyltransferase. All right? It actually makes sense because this is phenyl, this is ethanolamine. Okay? We transfer methyl onto a nitrogen. It actually works if you think about it. Right. What is the donor of the methyl group? Where do we get it from? There aren't that many donors of methyl groups in the metabolism. Okay, what does SAM stand for? Okay, the important bit. S-adenosyl methionine. Okay, it's a derivative of methionine. It's an S-adenosyl methionine. Okay, what is the other source of methyl groups in metabolism? We need it, for example, to synthesize nucleotides, thymidine, all sorts of things. Very good, okay? Methylmethylene tetrahydrofolate, so folic acid derivatives. That's the other big group of methyl donors, okay? But in this case, it's S-adenosyl methionine. That's the donor of the methyl group. Good, so that's the synthesis of catecholamines. Now, how do we degrade them? How do we get rid of them? Once again, as with glutamate and GABA, the majority of catecholamines, once they've been released into the synapse, are taken up by other things, usually by the presynaptic neuron. Okay? So we remove catecholamines from the synapse by reuptake. They're taken up back into the presynaptic neuron, and then they are usually recycled for further neurotransmission. Okay? So that's how we terminate the signal. We suck them up back into the presynaptic neuron, and we end the transmission. However, we obviously need ways how to degrade them and completely get rid of them and excrete them. For that, we have two enzymes. Okay, what are they?
I can tell you the letters.
Okay. Okay, we have monoamine oxidase. And we have catechol O methyl transferase. Okay, two enzymes. Now, what do they do? First of all, let's take monoamine oxidase. Monoamine oxidase, as the name suggests, oxidizes an amine. Okay? Where do we see an amine in these three molecules? Is there an amine there? Yes, here, here, and there's a secondary amine here. So these amino groups are oxidized and by that removed by monoamine oxidase. Okay? What is going to be the result? What's going to be the product of the reaction? Instead of an amino group, we're going to end up with an aldehyde. Very good. Okay? So instead of this, you make this. Okay? We chop the amino group off and we make an aldehyde instead. That works for all three. For dopamine, for noradrenaline, for adrenaline, it works exactly the same way. So monoamine oxidase just chops this away, makes an aldehyde. Now once we have an aldehyde, what usually happens in the metabolism? If an aldehyde, what do you do with this? You oxidize it, okay? So you oxidize it into a carboxyl group. And we have a carboxylic acid which is pretty well soluble in water and can be excreted. Okay? So that's what monoamine oxidase does. How about catechol O-methyltransferase? Again, it's in the name of the enzyme, what it does. What's catechol? Okay? This is catechol. So what will catechol O-methyltransferase do? It will transfer a methyl group to where?
The OH group that we added in the previous step.
Very good. Okay? So it's an O-methyltransferase. So unlike N-methyltransferase that we saw before, which put a methyl group onto nitrogen, this one puts a methyl group onto an oxygen. And it's this oxygen there, or this one here, or that one there. Okay? So monoamine oxidase takes the amino group, chops it away, turns it into aldehyde, and we can then oxidize it into carboxylic acid. Catechol O-methyltransferase takes a methyl group from where? From SAM, yeah, from S-adenosyl methionine, and puts it onto one of the hydroxyl groups into on the catechol ring of catecholamines. Now, the combination of the two produces the major metabolite of noradrenaline and adrenaline, which is called vanillyl mandelic acid. Now, I'm just writing this so that you see the name. It's used in clinical medicine to measure the production, in certain conditions, the production of catecholamine, so you can measure it in the urine. And it's actually because there are some tumors that can produce adrenaline and noradrenaline, so you can detect those tumors by measuring the amount of vanillyl mandelic acid in the urine. You don't need to remember the name. Just be aware that there is a product of the degradation that we can measure, and we do measure in the urine. Okay. Any questions about the metabolism of catecholamines? If not, let's have a 5-minute break. Right. me just have a snarky comment that now maybe you see that why we wanted you to, that's okay, yeah. Why we wanted you to learn all the metabolisms of amino acids, because they actually make these things a lot easier. So if you know how normal amino acids are metabolized, you know, remembering and understanding this is actually it's trivial, okay? Anyway, do you have any questions about the first half of the lecture?
Did you repeat the catecholamine transferase? What did it actually do?
Say again?
The catecholamine transferase, what did it actually do? It attaches...
You mean the catechol O methyl transferase?
Yes.
Okay, it attaches a methyl group onto one of the hydroxy groups on the catechol ring. One of the hydroxyl groups on the benzene ring. Okay? Just puts a methyl there. So it makes a methoxy group. Yep.
And what does that do for us for the degradation of the...
First of all, both the enzymes inactivate the catecholamine. Okay? So they stop being neurotransmitters. The other thing, monoamine oxidase makes them more soluble in water, so they can be more easily excreted. Any other questions? If not, let's move on to the next neurotransmitter. It's going to get easier and easier now. The next neurotransmitter is acetylcholine. How do we make acetylcholine? Add choline to acetyl. Okay, that's a good way of doing it. Okay, what is choline? What does it look like? With three what?
Carbon.
3 methyl groups. Positively charged. Okay. Okay, this is what it looks like. This is choline. Now, well, okay, this is choline, okay? So how do we make acetylcholine from that? CH3, 3 times, okay? Let me make it clearer. How do we make acetylcholine from that? What do we add? Sure. Do we just find an acetyl group lying around? No. Very good. Acetyl-CoA. And where do we attach the acetyl group? Okay, so we make what? What is going to be, what is? An ester bond. Very good. And we have acetylcholine. Right. How do we make choline? Does it look like something that you might be able to make from something that we already have in the body or something like that? It's not a vitamin, okay? To be fair, the brain actually takes up most of choline from the blood. It doesn't actually make a lot of choline. It's the liver that makes the most choline for the body. But still, it's something we make in the body. So when you look at the structure, is there something we could use to make it? Very good. We start with serine. Okay, this is serine, the amino acid. I'm sure you've met before. what do we need to do with serine in order to get this? Yeah? Very good. The first step, we decarboxylate it. And we get something like this. Okay, this is what we get by decarboxylation. What do you think it's called? What do you think is the name of this? We've already seen it before, huh? No, not the enzyme. I don't care about the enzyme. Well, it is serine decarboxylate, but what is the name of the compound? Very good. It's ethanolamine. Okay, ethanolamine. Remember, we saw it was part of the catecholamine molecule. Okay, so this is ethanolamine, and ethanolamine is also part of phospholipids. You probably remember that from your lectures on phospholipids. Phosphatidylethanolamin is one of the phospholipids. Okay, so from ethanolamine, we make choline by triple methylation. So we methylate it 3 times. What do you think is going to be the source of the methyl group here? Very good. You're in luck. It is indeed as adenosylmethionine. And we make choline. Okay? From that, we make acetylcholine as we established. How do we break acetylcholine down? You've already heard about that. We have acetylcholine esterase, and we break it down to what? What do you think we're going to break it down to? We can't break it into acetyl. Acetyl doesn't exist. Huh? We're not going to break it down to acetyl-CoA because that would need some ATP. We're going to break it down to acetate, to acetic acid. Okay? So we're going to get acetate and choline. Now, choline is taken up back into the presynaptic neuron and is recycled into acetylcholine and repackaged into secretor vesicles. And acetate is just plain metabolized by any cell that's around because metabolizing acetate is pretty easy. Yep?
I had a question about the, let's say I'm in folate. Are there a general group of reactions that would use?
No, there is really no general group of reactions. You pretty much have to remember which one's which. Yeah. So in nucleotide metabolism, it's all tetrahydrofolate. Yeah. There are specific reactions that have tetrahydrofolate, so yeah. Okay. And this is the metabolism of acetylcholine. Yes?
You only says serine gets what's it can be concentrated to again? Just the name.
To ethanolamine. Now, obviously, ethanolamine is not a systematic name. Okay, systematically it would be hydroxyethylamine, but ethanolamine, that's how everyone in biochemistry calls it. Do you have any question? The last step, acetylcholine esterase breaks down this ester bond. It's an esterase. Okay, breaks down this ester bond and makes acetic acid and choline. And that's taken up by the presynaptic neuron and is recycled into acetylcholine. Okay? Good. Any more questions about acetylcholine metabolism? I still see a lot of faces looking at me like, what the hell is he talking about? So, if you don't understand something, just ask. That's why I'm here. Otherwise, you could just watch the video.
Do we need to know the structures for the test on Monday?
I think they will be helpful. I think they're helpful, yes. You won't be asked to draw them, but they'll be helpful.
And these are all in the presynaptic clot, or not clot, just the presynaptic terminal? They're synthesized location?
Yes. All the neurotransmitters that we've talked about now, well, it's complicated with glutamate and GABA, but they're all synthesized in the nerve terminal, okay? The only exception are peptides and proteins, and we're going to mention some of them in a bit. But you actually remind me of something, and that's acetylcholine esterase is present in the synaptic cleft. Okay, so it's an extracellular enzyme. It's present in the synaptic cleft, and it degrades acetylcholine that's in the cleft. With monoamine oxidase, that's an intracellular enzyme, and it's actually an enzyme that's present in mitochondria. It's a mitochondrial enzyme. Monoamine oxidase is a mitochondrial enzyme. It's in the outer mitochondrial membrane. Catecholomethyltransferase is also an intracellular enzyme, but it can be secreted by the neurons. So it can be sometimes present in the extracellular fluid as well. Okay, so that's just about the location of these enzymes. Yes?
What's the enzyme called that produces acetylcholine? The one before the esterase?
Oh, it's called choline acetyltransferase. Okay? Choline acetyltransferase.
There's no energy used for this synthesis? Any ATP is used?
Okay, does anyone have an answer to, is there no energy used for the synthesis? Okay, acetyl-CoA itself has enough free energy in order to power the synthesis. The same way as it does in the Krebs cycle when forming citrate. Okay.
Which cell supplies the acetylcholinesterase?
It's in the presynaptic, it's all in the presynaptic neuron. Sorry, in the acetylcholine, oh, it's the postsynaptic cell. Okay, so it puts it on its own membrane. Yeah. Right, let's move on to the next neurotransmitter. And that's serotonin. Now, serotonin is sometimes called by a name that is more descriptive of its structure, and that's 5-hydroxytryptamine, or abbreviated as 5-HT. Okay? And when in the next lecture we talk about receptors, 5-HT is the abbreviation for serotonin receptors. Okay? So the real name for serotonin is 5-hydroxytryptamine, or abbreviated as 5-HT. So it's 5-hydroxytryptamine. How do you think we're going to make it? What are we going to start? Okay, start with tryptophan. Okay, start with tryptophan. You don't have to draw the structures. They're in any biochemistry textbook, okay? Or you know them already. Okay, so we start with tryptophan. How do we make 5-hydroxytryptamine? What do you think? There are probably going to be at least two processes, or two. Okay, we need to hydroxylate it. And? Transamination. Decarboxylation. Okay, so from an amino acid, we're going to make an amine. Okay, so correct, the two steps are hydroxylation and decarboxylation. Hydroxylation is the first one. The enzyme is called tryptophan hydroxylase, okay? And we hydroxylate the position 5. Well, obviously, it's a 5-hydroxytryptamine. 1, 2, 3, 4, 5. Oops, yep. So what do we get from that? From this reaction? Start with tryptophan, we added a 5-hydroxy group, so we have 5-hydroxytryptophan. And in the next step, we get rid of the carboxyl group, as you correctly said. Okay, what do you think the enzyme's called? Okay, yeah, you could call it 5-hydroxytryptophan decarboxylase. But in fact, this enzyme, and I should have mentioned it before, this enzyme is called aromatic amino acid decarboxylase. And it's the same enzyme that does the decarboxylation in catecholamine synthesis. Okay, so it's called L-aromatic amino acid decarboxylase. Okay, and it's a general enzyme that can decarboxylate. Is there anything you don't understand?
No, I'm just having trouble seeing. Does it say O2 on it or what?
CO2. Okay, CO2. L-aromatic amino acid decarboxylase. Okay, it's the same enzyme as dopa decarboxylase. Okay, but you can call it basically whatever you really want in this case. Right, I'm not going to draw the structure of serotonin or 5-hydroxytryptamine because it should be obvious. We just remove the carboxyl group. Okay, and we get an amine, and that's serotonin. Okay?
So the next step of that
is serotonin. Yeah. Yeah. How are we going to degrade it? I don't expect you to know it, just, you know, try something.
Catechol-O-methyltransferase?
Can we use catechol-O-methyltransferase here? Well, there is an OH group. There's no catechol there. Okay, catechol is a benzene ring with 2 hydroxyls. Here we have actually an indole group with a hydroxyl. We can't use catechol-O-methyltransferase, but we can use.
Is it pumped back to the presynaptic?
Yes, yes. Thanks for reminding me. Yes, serotonin, once it's released into the synaptic cleft, is taken up back into the presynaptic neuron. Okay, so that's again the same thing as for catecholamines is the same for serotonin. It's reuptaken to the presynaptic neuron. But then we also need to get rid of it at some point. So we can't use COMT, but we could use. Okay, we can use MAO, monoamine oxidase. Okay, because we do have an amino group here. Sorry, it should be plus. We do have an amino group there. So we can use monoamine oxidase. And we can oxidize 5-hydroxytryptamine into first an aldehyde and then into carboxylic acid. Same way as with catecholamines. Okay? The final product is called 5-hydroxyindole acetic acid. Again, it can be measured in the urine as a marker for serotonin production in the body. That's all you need to know. Okay? Do you understand how monoamine oxidase will...
Also measured with that vanilla mandelic acid?
Well, vanilla mandelic acid is the end product of catecholamine degradation. Well, specifically adrenaline, noradrenaline. 5-hydroxyindole acetic acid is the measure of degradation or the production of serotonin. Okay? So yeah, they're similar markers, but they measure 2 different things.
Can you write it down?
I can. (5-hydroxyindole acetic acid). Okay? 5-hydroxyindole acetic acid. Yeah? You don't need to remember that. Okay. Any questions about serotonin? Yes?
You were saying something about MAO.
Monoamine oxidase, yes.
Yeah.
Yes, I was saying that monoamine oxidase, in addition to degrading catecholamines, it can also degrade serotonin and make 5-hydroxyindole acetic acid. Okay? Good. Moving on. There is another way to not really degrade serotonin, but to turn it into something else. And that's the synthesis of a different hormone called melatonin. Melatonin is synthesized from serotonin. And I'm just going to say that it's synthesized in 2 steps. One is that we add an acetyl group here onto the amino group. So we add an acetyl group here. We make an amide. And then in the next step, we add a methyl group onto the hydroxyl. And that makes melatonin. Don't worry about it, okay? Melatonin is synthesized from serotonin in 2 steps. All you need to know, okay? Those of you who want to look it up, it's everywhere, okay? But I'm not going to go into the individual steps because there's really no point, okay? Now. Good. So let's get to the next neurotransmitter, which is histamine. Now, as promised, it's going to be getting easier and easier. Histamine. Any question? What do you think is going to be the starting material for histamine? Histidine. Very good. How are we going to make histamine from histidine? By decarboxylating it. That's it. Okay, we have histamine. Now, the enzyme here is not aromatic amino acid decarboxylase because histidine is not an aromatic amino acid. Okay, it's called histidine decarboxylase. The degradation of histamine is not done by monoamine oxidase because there are actually 2 amino groups, 2 sort of amines in histidine. So there is an enzyme called diamine oxidase. Now, I'm going to put that in brackets. You don't need to know that, okay? But there's a different enzyme. What you do need to know, it's not metabolized, it's not degraded by monoamine oxidase, okay? There's a different enzyme for that, which is diamine oxidase. Yes? Diamine oxidase degrades histamine. Okay? In a similar way as we saw with monoamine oxidase, just a different enzyme. Okay? That's all for histamine.
Can you go back to melatonin real quick? And
Yep.
How is it degraded?
That's a very good question that I got from the Czech students as well. I don't know. I think it's excreted either completely unchanged or just glucuronidated. I don't think there is a metabolic pathway for chopping it into bits. But honestly, I just don't know. Okay? I don't know.
On that one, you said the OH group was
Methylated.
yeah, and the other.
Okay, let me just. I thought he wouldn't be interested, so I wasn't. Okay, this now is serotonin. Can you see that? Can you see this is serotonin? Okay, so what we do, the 2 steps that we do to make melatonin, first of all, we add an acetyl group to the nitrogen. Okay, so we make N-acetyl serotonin. And then in the next step, we methylate the hydroxyl group. And this is now melatonin. Okay? So you wanted to know, now you know. Right. I'm actually doing pretty well. The next group of neurotransmitters that I want to talk about are peptides, peptide neurotransmitters. As I said before, unlike all these small molecule neurotransmitters, peptides are synthesized in the cell bodies because they need the nucleus and they need the transcription apparatus that's in the cell body. Now, the majority of peptide neurotransmitters are synthesized as huge long proteins that are then chopped into bits that are the actual neurotransmitters. Okay? So there are several relatively large peptides that are then processed by chopping them into small bits. Now, I want you to remember one of them, which is quite important for several different things. The abbreviation is POMC. Does anyone know what it stands for? It stands for pro-opiomelanocortin. Pro-opiomelanocortin. And I'll now explain why this horrible, horrible name. This horrible, horrible name is, it's so horrible, because it gives rise to several different peptides when it's chopped into bits. And the three of them, one is beta endorphin, which is an opioid. Another one is a very strange signaling molecule called melanocyte-stimulating hormone, which in some animals activates melanocytes to basically shift pigment around. Okay? All right, melanocyte-stimulating hormone. And another bit that can be formed from propiomelanocortin is the adrenocorticotropic hormone, which you've heard about. So that's the cortin. Okay, it's adrenocorticotropic hormone. Okay, so propiomelanocortin is a big precursor. There are actually several other peptides or alternative, peptides that can be formed from that. These are just the three important ones, and also the three that explain why this name is formed in such a way. Okay, so we can get some opioids from it. We can get some hormones that do something with melanocytes, and we can get some hormone that does something with adrenal cortex. Okay, that's all I'm going to say about peptide, about the synthesis, and well, the degradation is just that the peptides are chopped up into amino acids. Okay, nothing special there. Do you have any questions?
What was the MSH?
Melanocyte-stimulating hormone. Okay, melanocytes, yeah, melanocyte-stimulating hormone.
One of those that makes a hormone, and it's called pre-pro. I think it might be insulin.
Yes.
Pre-pro-insulin.
Yeah.
What's the pre? I guess the pro bit is before the.
Okay. Does anyone know what the pre bit is in pre-pro insulin?
The location of where it is.
It's the signal sequence in the beginning that directs the protein that's being formed into a correct place in the endoplasmic reticulum.
So what's the difference between the pro bit and the pre bit?
The pro bit means it's unfinished. We need to splice it to get the final product. The pre bit is the signal sequence.
But what's the point in the pro if it didn't do anything?
It just says it's a protein that is going to be chopped up into the real product. Okay, so what he's saying is, why isn't there opiomelanocortin?
Yeah.
Is that your question?
Yeah.
Okay, well that's how it is. Okay, pro-opiomelanocortin is spliced into these products, not into opiomelanocortin. It's just a naming thing. Okay. Yeah. Okay, finally, we get to, well, we still have 3 more groups of neurotransmitters, but we're going to be very quick. Okay, it's gonna be quick. Don't worry. The first one is nitric oxide. And you already know about that, so it's going to be super quick. How do we make nitric oxide? From arginine. What are the products? Citrulline and nitric oxide. What are the enzymes? So what is the enzyme? NO synthase. Do we have different types of NO synthesis?
Yes.
Yes, like, for example? 1, 2, and 3. Okay, that's interesting. That's what the Czech students said as well. Indeed, we can call them 1, 2, and 3, but there is actually a better name of naming them. There's enos, ennos, and inos. E stands for endothelial, so it expressed in endothelial cells. N stands for, okay, it's the neuronal one. And I stands for inducible. You can also think that the I stands for immune cells, because the inducible NO synthase is present mostly in immune cells. Inducible. So these two NO synthases are together called constitutive, because they are present in the cell all the time. They're constitutively present in the cell all the time. And they need to be activated in order to start producing nitric oxide. What is the activation signal for constitutive NO synthesis? Do you know? Okay, acetylcholine, that's true, but that needs to go through some more steps. What's the actual signal for activating NO synthase? Calcium, okay? They're activated by calcium. The inducible or immune nitric oxide synthase is not constitutive. It's not in the cell all the time. It needs to be induced. It needs to be produced when it's needed, okay? But once it's produced in the cell, it's not really regulated very much. So once it's there, it produces nitric oxide. So that's the difference between the constitutive ones, which are there all the time and only become activated when calcium rises, well, calcium levels rise. The inducible one needs to be produced as a protein when the cell needs to produce nitric oxide. Okay? Do you see the difference between inducible and constitutive enzymes? Okay, most people do. That's good. Okay, how is nitric oxide degraded?
It's made into nitrate.
Nitric oxide is degraded by oxidation first into nitrite and then into nitrate. Do you know what catalyzes this oxidation?
Buffer system in the blood.
It is a buffer system in the blood. But which one? No, it's actually hemoglobin. Okay? Oxyhemoglobin contains oxygen. It has fairly high affinity for nitric oxide, so nitric oxide binds to oxyhemoglobin and is oxidized into nitrite. Okay? And nitrite can be just excreted in the urine. You can measure nitrite excretion in the urine, which tells you about how much nitric oxide people produce. Okay? It can also be oxidized to nitrate, which is actually done by bacteria, etc. Not that important. Okay? So nitric oxide is degraded to a large extent by oxyhemoglobin into nitrite, and then potentially to nitrate. Okay? Last two things. Cannabinoids, endocannabinoids. We're not going to spend a lot of time with cannabinoids just to say, well, obviously, delta-9 THC, tetrahydrocannabinol, which is in marijuana, needs to have some receptors to bind to. It binds to cannabinoid receptors, and these receptors were obviously not made for THC, they were made for endocannabinoids, some ligands in the brain that can bind to cannabinoid receptors. One of these ligands, and the only one that actually has been studied and confirmed to be a natural ligand of endocannabinoid receptors, is called anandamide. Anandamide, which is a name, well, obviously it's an amide. And ananda, I'm told, in Sanskrit, means bliss. Okay? So that's just the formation of the word. Okay? Bliss, like elation. Yeah. What other words I could use? Some native speakers tell me some other synonyms of bliss. Anyway, okay, so it's just the name is formed from a Sanskrit word for, you know, being high, and it's an amide. Now, why am I talking about it? I'm talking about it because it's chemically a very interesting neurotransmitter, because it's actually formed from fatty acid. So anandamide is actually a compound from arachidonic acid plus our favorite ethanolamine. So you take ethanolamine, you react it to make an amide bond with arachidonic acid, and you have anandamide. Okay? That's all you need to know, that it's a fatty acid derivative from arachidonic acid. And final word is about purines. And that's just to say that certain purines act as neurotransmitters or co-transmitters or neuromodulators. ATP is one, so ATP can actually act as a signaling molecule. Okay? And also adenosine. There are specific receptors, purinergic receptors, that actually react to these signals. I'm not going to talk about the synthesis or the degradation because you've had that in nucleotide synthesis and nucleotide degradation. So you should know that. So this was the very last thing. Do you have any questions?
What is their effect as neurotransmitters?
There are all sorts of effects. There are all sorts of receptors. We're going to talk a little bit more about them in later lectures, a little bit more. Okay.